Hello everyone, my name is Delian and I would like to give you a quick recap of our review of in vitro models of medulloblastoma. The purpose of this paper is to systematize the available information on medulloblastoma models. We've updated cell line information to include subtype and the evidence behind it, and have presented strategies to improve the physiological relevance of in vitro models by using 3D cell cultures, co-cultures with the normal brain, and the right extracellular matrix. Medulloblastoma is the most common malignant brain tumor in childhood. Despite improvements in overall survival, multimodal therapy has serious side effects such as decrease in IQ and reduced independence for medulloblastoma survivors. That is why we need a rational approach to identify safer therapies. Medulloblastoma is no longer viewed as a single disease and is now split in 4 molecular subtypes. 10% of patients belong to the Wnt subtype and carry a good prognosis with over 90% 5-year survival. 30% of patients present with mutations in the Sonic Hedgehog pathway and carry an intermediate prognosis, as do patients from the largest group of medulloblastoma, group 4. At the other end of the spectrum, patients from group 3 often present with metastases and have poor survival. Splitting a rare disease like medulloblastoma in multiple subtypes poses problems for clinical trials as there are a small number of patients. Therefore, patients' tumors would need to be faithfully represented in the laboratory before treatments are trialed in patients. This means that we need preclinical models which match patients' tumors as closely as possible. Cells from patient tumors can be cultured in a laboratory, and the first culture is called primary. As the cells expand and are transferred to new flasks, they can form an established cell line. Established cell lines can be injected into the brain of immunocompromised mice to form cell line xenografts. Another way to model medulloblastoma before clinical trials is to culture cells directly from the patients into the brains of immunocompromised mice and form patient-derived xenografts. Alternatively, researchers can genetically modify mice to activate oncogenes or delete tumor suppressor genes to create artificial cancer models called genetically engineered mouse models. While this review briefly touches upon in vivo models such as patient-derived xenografts and genetically engineered mouse models, its main focus is on in vitro models. Cell line models are readily available. They rest upon 30 years of research, and the results derived with cell lines can be easily replicated all over the world. Nevertheless, cell cultured in artificial solutions on plastic in the laboratory can adapt to culture conditions and can change with time. This word cloud shows the number of literature citations for each medulloblastoma cell line divided by the number of years the cell line has been around for. It shows that the most popular cell line is Dowie, with over 2,500 citations for the past 30 years. In total, researchers have published on 44 medulloblastoma cell lines. However, only 18 of those have been subtyped to belong to a single group. 3 may belong to group 3 or 4, and 23 cell lines have not been subtyped at all. While tumors with MYC amplifications are seen in only 4% of medulloblastoma patients, there are 11 group 3 cell lines with MYC amplifications. Some cell lines lie somewhere between group 3 and 4, such as the second most popular cell line D283. If we look at the bigger picture and compare the frequency of certain types of medulloblastoma tumors in patients versus the number of available cell lines, we can see that tumors with MYC amplification and mutations in p53 are overrepresented in cell lines. There are many genetically engineered mouse models which cluster close to Sonic Hedgehog tumors, but none for group 4. Critically, patients with Wnt tumors, group 3 tumors without MYC amplification, and especially group 4 tumors are underrepresented by preclinical models of medulloblastoma. To faithfully model tumors in the laboratory, we need not only the right cells, but also the right environment. Cells in patients grow in 3D, not as a carpet on plastic. Similarly, they interact with the normal brain, and including the normal brain in tumor models can not only give an indication of drug safety, but would also increase physiological relevance. In addition, the extracellular matrix can modulate cell signaling and plays an important role in metastasis and resistance. Future in vitro models need to incorporate brain-relevant extracellular matrix based on hyaluronic acid and reduce the emphasis on basement membrane-derived matrices containing laminin, collagen, and fibronectin, which are not a major part of the brain extracellular matrix. To recap, we need to characterize and classify the remaining 23 established cell lines. We need to focus on developing in vitro models for Wnt and group 4 tumors. We also need to improve the way we describe old and new cell lines and update this information as soon as new developments emerge in the field. This can be accomplished with a centralized database and a linked cell bank of well-characterized lines with molecular characterization and drug sensitivity information. For all cell lines and models, we need to ask the question, which patient tumors does this represent, and how useful is it in answering my questions? The take-home messages are that we need models which can be matched to patients and the degree of similarity quantified. Furthermore, a central repository for well-characterized cell lines and a database for their characteristics would enable researchers to select the right tool for their work. And beyond genes, culture conditions such as 3D, co-cultures, and the extracellular matrix can influence phenotype and improve the physiological relevance of in vitro models.